Hello YR9s, in this video I'm going to go through the cell structure and transport end of topic test. Watch the video and make the corrections in your test paper using green pen and hand it back to your teacher. So question 1. Living organisms are made of cells. Animal and plant cells have several parts. Each part has a different function. Draw one line from each part to the correct function. So cell membrane is to control the movement of substances in and out of cells. Mitochondria is where aerobic respiration takes place, where most energy is released in respiration. It could be that. Nucleus controls what goes in and out. Now, where proteins are made, that will be ribosomes. Write this in green pen in your test paper. Figure 1 shows some parts of a plant cell. So you have a plant cell with the cell wall, cell membrane, cytoplasm, mitochondria, and ribosomes. Name 3 parts of a plant leaf cell that are missing from figure 1. So the main would be the nucleus and a permanent vacuole. And the third is the, what's missing? Chloroplast. Next question. What is the function of the cell wall in a plant cell? The function is to provide support or strength. You could also say to protect the cell as well. Which 2 parts in the diagram above are not found in an animal cell? The 2 parts would be cell wall you could also say chloroplast or the vacuole. Any 2 of these. Question number 2. Root cells absorb substances from the soil. Figure 2 shows the concentration of different substances outside a root cell and inside a root cell. So you have substance C, which is these big circles, and the small dark dots are water, and the smaller circles are the potassium ion. So let's have a look at the question. The cell membrane of a plant cell is partially permeable. What does partially permeable mean? It means to allow some substances to go through. Partially means part of it. Next question. How does water move into root cell in figure 2? That would be by osmosis. How does potassium ion moves into a cell? That would be by active transport. Explain why substance C in figure 2 does not move into the root cell. So looking at substance C, which is the big circles, they are too big to fit through this gap, hence it can't actually move into the root cell. So substance C is too big to pass through the cell membrane holes. Next question. A student investigated the effect of different concentration of sugar solution on the size of potato cubes. This is the method used. Cut one potato cube, record the mass of the potato cube, place the potato cube into a beaker of concentrated sugar solution. After 1 hour, record the mass of the potato cube again. Repeat steps 1 to 4 using very dilute sugar solution. Give 2 factors that the student should control in this investigation. So you could say the size or shape of the potato cube. You want to keep that same, because if you change the shape or the size, that will affect the surface area. The time, the potato cubes were left in sugar solution. Next question. What piece of equipment could the student use to accurately measure the mass of each cube? You need to say weighing scale, not simply scale. Weighing scale, or you could say top and balance. A potato cube is placed in a concentrated sugar solution. What will happen to the mass of the potato cube? Now, if you place it in a concentrated sugar solution, so if I draw a very simple diagram, that's the solution, and let's say it's very concentrated, and that's the cell, and that's a lot of sugar particles outside than the inside. Then the water will actually move out of the cell, so which means the potato cube will decrease in mass. Question number 3. Substances can move into and out of cells by these 3 processes. The diagram shows the concentration of different substances inside and outside a root hair cell. How would each substance move into the root hair cell? Draw one line. So you can only draw one line for each. So water molecule, nitrate ion, and the third is also nitrate magnesium ion. So osmosis is the movement of water molecules only. So that would be osmosis. Now, diffusion is from high concentration to low concentration. So high concentration nitrate ions to low concentration. So that would be diffusion for the nitrate ion. And active transport is from low concentration to high concentration, or against a concentration gradient. So that would be magnesium ions. The image below shows an epithelial cell from the lining of the small intestine. In the image above, label part A. Part A is going to be the nucleus. So how are most soluble food molecules absorbed into the epithelial cells in the small intestine. That would be by diffusion. Suggest how the highly folded cell surface helps the epithelial cell to absorb soluble food. It increases the surface area. Next question, C. Epithelial cells also carry out active transport. Name one food molecule absorbed into epithelial cells by active transport. That would be glucose. Why is it necessary to absorb some food molecules by active transport? The reason it has to absorb food by active transport is because it's trying to move it against a concentration gradient. So, to move molecules against a concentration gradient. 3. Suggest why epithelial cells have many mitochondria. So remember, mitochondria is where energy is produced. So active transport requires energy, and that energy is going to come from respiration in the mitochondria. So requires energy from respiration. Next question. Question 5. An understanding of relative size is essential in science. The diagram below shows two model cells. Describe how the surface area to volume ratio. So if I say SA for surface area and V for volume, ratio changes as the length of the side of the model cell increases. So if I label that as cell 1 and cell 2, you should include calculations. So for cell 1, remember to find the surface area, you find the length times the width, multiply that by 6 because it has 6 sides. Volume is given by length times the width times the height. So we are going to use these two equations. So cell 1 surface area over volume. So surface area is going to be length is 0.5 times 0.5. Multiply that by 6 sides. And volume is going to be 0.5 times 0.5 times 0.5 times again. So let's find what these values are. So for surface area, it'll be 0.5 times 0.5 times 6. That gives me 1.5. 1.5. Divide that by 0.5 times 0.5 times 0.5, which is 0.125. So Let me choose a different color for that. Fine. So surface area to volume ratio is 1.5 is to 0.125 for this cell 1. So for cell 2, we do the same thing. Surface area over volume. Surface area is going to be, each side is 1 centimeter, so 1 times 1, multiply that by 6, that would give me 6. Divide that by 1 times 1 times 1, that is also going to be 1. So 6 over 1. So surface area to volume ratio is 6 is to 1. Now the question is asking, describe how the surface area to volume ratio changes as the length of the side of the model cell increases. So surface area to volume ratio increases. So it's gone from 1.5 to 6 and from 0.125 to 1. So surface area to volume ratio increases as the length of cell increases. And that's 1 mark there for that, 1 mark for this, and 1 mark for this. And that brings us to the end of the questions. Yeah. Watch the video, make the corrections in green pen, and bring the paper back to class. Thank you. I will see you in the next video.